For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
Okay, looks like we are live for another great show, guys. This is another great show this week. It's a busy one, but we've got another awesome guest with us today. I want to thank Dr. Rob Stadler for once again giving us his time for this incredibly important discussion on abiogenesis, but specifically on his newest must-read and must-have book, The Stairway to Life, An Origin of Life Reality Check. Rob, thanks so much for joining us today.
Hey, thank you for having me. It's great to be here, and I appreciate you. You haven't gotten tired of me yet, I guess. This is the third time, huh?
No, you're a fan favorite. They'll probably kick me off the channel before they kick you off the channel, Rob. I want to point out to the audience, please, if you have not yet, please check out Rob's previous videos with us. We've had him on to debunk Professor Dave. That's a must watch. And we've recently had him on at the end of 2020, actually, to discuss his book on evolution. And that was an extremely informative and helpful interview. So actually, I want to thank George. George is going to be joining us for this interview today as well. George, thank you for being here.
No problem. I was encouraged by my first joke to Rob. He laughed at it. So I'd like to maybe tell him another one, either now or later on. It's up to you.
You can start it off now, George, if you like.
All right, Rob. What was the biologist wearing on his first date with a hot chick? Designer jeans.
Oh, designer jeans.
I'll stop there then.
I can't top that, George. So no
don't put your day job, George.
Good
No
on you,
raise
Rob.
for you, George. Anyways, I want to point out before we get this party started here to the audience. Good to see everybody. Good to see so many brothers and sisters. I'm really excited for this. Please tag me with your questions. We're going to keep it on topic and keep it relevant to abiogenesis and specifically Rob's book, The Stairway to Life. So tag me with any questions. If it's any questions that pertain to something Rob's discussing in the presentation, then I can stop him and we can kind of clarify from there. Or else I'll save them for the end of the presentation where we will have a short audience Q&A. So anyways, enough from us. I'm going to hand it over to you, Rob, for some brief words in terms of introduction, and then we can get into your presentation.
Well, today we're addressing, I think, one of the most important questions you can ask, which is, how did life get started? And if you've been told that life started from purely natural processes, just chemistry and physics doing what it does, then this show is for you. So I hope you stick with us and keep an open mind.
Amen. I agree, Rob. It's one of the most important topics. And I got to say, ever since we scheduled this, I've been extremely excited. So once again, thanks for giving us your time. And would you like to get right into the presentation?
Yeah. So I have some slides that we'll go through, and yeah, like you said, we can take questions at the end or jump in if you feel the need to. But I can start to share my screen here.
Sure, I'll get that going for you as well. Here we go.
Hopefully that's pretty clear. Is that good?
Looks good. Looks great.
So here's the book we're talking about today. And I want to point out my co-author is Change Tan, and she's a professor from the University of Missouri. And she and I got very passionate about this topic and to write this book because it just seemed like that there's an increasing barrage, a kind of a media blitz going on about natural explanations for life. It seems like every day you see something on the internet that's a new theory, new data, all this that's always very encouraging about natural causes behind life. And so we felt the need to provide a reality check, and that's what this book is all about. We strongly believe that science not only doesn't support natural causes, but actually quite points the opposite direction. So that's what this is all about. So I'd like to start with something that we can all agree on, all of us can agree on, that when a living thing dies, right, any living thing is going to die. And when it dies, there is a one-way process, and it's decay and disorder, whereby the very complex structure, the very complicated molecules, all the information content that was in that living thing decay, begin to fall apart. They lose their organization, and it degrades down to very simple, basic molecules that are left after that process. Now, of course, this is blatantly obvious. It's a natural process. This is where physics and chemistry and thermodynamics take us. It's directly observable. We've all seen it. And not only have you seen it, but you've seen it repeatedly. And it's universal, meaning it happens every time, always, to every form of life. And it's irreversible. Once you start down the path, you can't just turn the path around and go back to life. And all this is common sense. And so it becomes quite disturbing to me, actually, that a growing number of people, in fact, some very smart people, actually believe that quite the opposite has happened at least once in history, where you have, some sort of primordial soup on the earth before there was any life, or even on some other planet before there was any life. And somehow physics and chemistry performed the exact opposite, where you have very simple molecules coming together, becoming more and more complicated, becoming organized with each other, developing great depths of information content, all contained within a membrane, and you have life. And this is called abiogenesis. And that's what this is all about today. This talk is there any reality to this possibility here? And I can say up front that this is a very unnatural process, that as we saw, physics and chemistry, thermodynamics are always pushing down. There's a gravitational pull downward here, and not something that elevates this process. It is unobservable because, frankly, no one ever has observed this process. And that's kind of a hallmark of science. If you can't observe it, then what is your argument? It's unscientific then, because no one's ever observed it, and it's unnatural. And so I think it's irrational to hold this kind of a belief. And frankly, it's a faith-based belief. So I kind of scratched my head and wondering where this belief came from in the first place. So there's a little bit of history here, and some of this history will give you a feel for why I'm passionate about this. So back in the early 19th century, this man here wrote a book, and he said in that book, "There's therefore no absurdity in believing that the most simple animals and vegetables, in other words, single microbes, single-celled organisms, may be produced by the congress of the parts of decomposing organic matter without what can properly be termed generation." So in other words, no parents involved. Somehow the simplest of single-cell organisms are just produced through decaying matter without any kind of parent. That's what he's saying. So any idea who this person is? It's quite important to know who this is. This quote came from Erasmus Darwin, who is the grandfather of Charles Darwin, and no doubt a big influence on Charles Darwin. And he strongly believed that life could spontaneously generate. And his book called The Temple of Nature, I think, is quite telling, because if nature is your God, you're going to believe this is true, and you write a book called The Temple of Nature where you worship nature. So back in the early 19th century here, single-celled organisms were kind of a novelty. Nobody really knew much about microbes. And so there was a belief when they were first discovered that they must be very, very simple organisms, because they're microscopic and they just looked like little blobs. In fact, here you have Ernst Haeckel later in the 19th century, and he gives a feel here for what was thought of single-celled organisms like this. So during late years, we've become acquainted with Monera. Monera here, you could basically substitute the word bacteria, because back then they called it phylum of Monera. These are organisms which are, in fact, not composed of any organs at all, but consist entirely of shapeless, simple, homogeneous matter. The entire body of one of these bacteria during life is nothing more than a shapeless, mobile little lump of mucus or slime, consisting of an albuminous combination of carbon. Simpler or more imperfect organisms we cannot possibly conceive. So, today, looking from 2020, with 2020 hindsight, you could say, we can see this as a statement of profound ignorance, knowing that even the simplest of bacteria is extraordinarily complicated. And to think it was just a little lump of mucus or slime is a very different viewpoint than we hold today. But if this was your belief back then, you can imagine that a simple little lump of mucus or slime could spontaneously generate from some decaying matter, right? So maybe believing that life started in that manner, wasn't so crazy back then when this was the mindset. But we know better today. We're smarter than that. We know a lot more about chemistry and physics and about biology, about living things. So we ought to know better. But unfortunately, we continue to see very naive comments like this being made by scientists and very influential people. For example, we can find George Gaylord Simpson, very prominent scientist in his time. Back in 1952, we had the Miller-Urey experiment here, where some simple gases were put into, some bubbling water and some electric sparks, and they produced some amino acids. And that was revolutionary. And about 8 years later, we have George Gaylord Simpson writing this in the journal Science. He said, the consensus is that life did arise naturally from the non-living, and that even the first living things were not specially created. The conclusion has indeed really become inescapable, for the first steps in that process have already been repeated in several laboratories. This, I believe, even though it's a good 80 years after the previous quote by Ernst Haeckel, this is also a statement of profound ignorance. To think that the production of a few amino acids makes it inescapable to believe that life arose naturally from the non-living, just because you have a few amino acids, it's incomprehensible to me that you could think a few amino acids means that you could produce life. And just because you've done it repeatedly by a few labs, I don't know, this quite shocks me. And I think that the title of his paper is also very telling, The World into Which Darwin Led Us. I think this kind of a profound stupor of this statement is in fact the world into which Darwin has led us to believe such things. Unfortunately, even though this was 60 years ago, the saga continues. We have Carl Sagan. Everybody knows Carl Sagan, a very prominent scientist. And he actually said the origin of life must be a highly probable affair. As soon as conditions permit, up it pops. Astounding, because there really is no empirical evidence to support this whatsoever. But even more astounding is that Carl Sagan is rather famous for also saying, extraordinary claims require extraordinary evidence. So this is an extraordinary claim, and I don't know of any evidence for it. But he made it because he believes that life just started naturally. Then we have a prominent scientist who used to be an MIT physics professor. Now he's at Georgia Tech. This is Jeremy England. He says, you start with a random clump of atoms, and if you shine light on it for long enough, it should not be so surprising that you get a plant. I just had to pause there. Just pause there for a moment of effect. So people are still making these statements. They're still grossly oversimplifying what is required for life in order to make it seem possible that physics and chemistry can produce life. Just a few more. Here we have Bill Nye, the science guy. He wrote a book and he said the origin of life just requires some raw material that could allow the spark of life to emerge. This is not a statement of science. This is kind of a statement of faith or fantasy. I think maybe he was a fan of Pinocchio, because in that movie, of course, there was a wooden puppet that's turned into a boy through the spark of life. Of course, he means a purely natural process here. And ironically, his book is entitled Undeniable. And then what provokes me the most, actually, is, I mean, people can believe what they want to believe, but to actually force it upon our students in public schools is what I find the most disturbing. So here's a popular high school biology textbook for public schools. And it says, life began when organic molecules assembled in a coordinated manner within a cell membrane and began reproducing. And another popular biology textbook just simply says, life arose from non-life via chemical evolution. And these are statements that don't even have a hint of uncertainty to them. There's no maybe or possibly or we think or we hypothesize. It's almost like they're slamming their fist on the desk, and saying with absolute confidence this is true. And it's deeply disturbing because to me the science is exactly the opposite. And yet these books are able to make such bold claims as this. So I think we need to start to look at the science then. What would it take to start life by purely natural processes? What would it really take? And this is where I think, my co-author Change 10 and I had a really strong synergy here because she's the biologist. I have some biology background, but also an engineer. And I kind of put an engineering twist on it. Like if you're going to build life, engineers know how to set requirements to get something done, to build something. So what would it take to build life? And we came up with this metaphor of a stairway where down here at the bottom, you have simple chemistry. You just have a few simple molecules like in the Miller-Urey experiment, and that's prebiotic. You don't have anything yet. And you're going to have to climb step by step. Each one of these steps is a requirement in order to get up to a living thing, the first living thing where you have biology going on instead of chemistry. So between chemistry and biology is this huge gap that we have to somehow span with purely natural processes. And I can tell you with certainty, this is a strong prediction, that in the years to come, as we learn more about what is required for minimal life, there will be new steps added to this stairway over time, steps that we don't know about yet. Because over time, the gap has been growing exponentially as we learn more about life. The gap between chemistry and biology is growing exponentially. And we also know that, as we already discussed, that death and decay is a kind of a gravitational force that's pulling you down the staircase, just like gravity does. And this is a very natural process. Chemistry and physics are going to pull you down here in a purely natural way to get back to very simple basic molecules after you die. And yet somehow people are trying to convince us that physics and chemistry somehow walks their way up the staircase and made it to life. And so we're going to go through some of these steps. We're not going to cover all the steps. You're going to have to, go to the book if you want more detail or ask questions. But I'm just going to pick out some of the highlights and go through some of the requirements to get to life. So let's start with the first one down here, which is the formation and the concentration of the basic building blocks. We're talking about amino acids and carbohydrates and lipids and nucleotides. These are the basic building blocks of all life. And so somehow you have to form these and you have to concentrate them. And so you'll find experiments, many of them, many published experiments, somewhat like Miller-Urey started by that concept here, where, yeah, they can produce amino acids, and yes, they can produce nucleotides in very small quantities and just under the right conditions. And so they kind of get lost in the trees and forget about the forest and claim victory for very little steps of progress toward this. But there are many things that are kind of cheating about all of these processes. And I'm just pulling out my top three here. The first one is that when they do this kind of origin of life chemistry, they're starting with a very small number of pure concentrated reagents. They go to the lab supply shop and they buy perfectly pure chemicals and put them in their perfectly pure glass container with pure water, whatever it is. It's all pure and clean. And they end up with a small amount of something like amino acid and they claim victory. The reality in a prebiotic world would be quite messy. We're talking about a very complex mixture of many different interfering molecules. And some evidence of that comes from the Murchison meteorite, which gets a lot of attention. a meteorite fell down to the earth and someone, looked inside of it and found amino acids. And so there's excitement there to think that out in outer space, something produced amino acids, which is really cool. But it's almost never mentioned that the meteorite also contained tens of thousands of different molecular compositions, likely millions of diverse structures in there, which suggests that the extraterrestrial chemodiversity is high compared to terrestrial relevant biological and biochemical driven chemical space. What this is saying is, for every useful biological molecule found in this meteor, there's a whole bunch of junk, a whole bunch of organic junk that is not of interest. It interferes and in fact may even be detrimental or poisonous to life. And that's what is to be expected, is a huge mess. So in this vial, instead of 6 pure chemicals, they should start off with tens of thousands of different organic compounds, a big mess, and then see what comes out of it. Recently, just last year, there was a publication of a computer simulation called Alchemy from a Polish group. And this is computer simulation, but it's very similar. They started with just 6 prebiotic molecules and let the computer go through 7 generations of what would come out of, chemical reactions from these 6 molecules. And what they came to was over 36,000 different molecules came out of it. And they noted that only 82 out of the 36,000 were related to or found in living organisms. So basically you have 36,600 junk molecules for every 82 that are actually related to life. And this will stifle any kind of useful progress, but you don't hear about that stuff. And finally, not only do you have to pull out the good molecules you want from all of this junk, somehow you have to concentrate it then, because concentration can indeed be seen as a chemical constraint in the origin of life, since chemistry cannot operate below a certain threshold of concentration. Some of you may have learned Le Chatelier's principle, where you have to have a concentration of the reagents in order to drive the reaction forward. So this is all just my first complaint about the Miller-Urey type apparatus. Second one is that each experiment addresses only a very small portion of the overall origin of life problem, and they address it in isolation. So the way to picture this is, let's say you have to make Thanksgiving dinner. A friend of mine gave me this analogy. I love it. You have to make Thanksgiving dinner. You got to make your mashed potatoes, your turkey, your stuffing, your green beans, the pumpkin pie, the whipped cream, the ice cream, the apple pie, the coffee. The complete Thanksgiving dinner has to be made. And so typically you do each one in isolation. But for origin of life, the reality would be you've got one pot and you've got to make the complete Thanksgiving dinner in that one pot all at once under the same conditions of heating or cooling or whatever. This is more of a reality of how you would have to do it. You can't do everything in isolation. And then finally, chemists doing these kind of experiments, they claim a victory for one-time production of a small amount of what they're trying to get, a few drops of amino acid. The reality would be, because origin of life is supposed to have happened over hundreds of millions of years, you need some type of technique that's going to do continuous production of a large quantity of the substance over great spans of time. So, let's imagine that you build yourself a chemical factory that cranks out laundry detergent, right? Of course, it takes a lot of intelligence to build that factory, and to keep it running takes a lot of intelligence and supply chain and maintenance and all that stuff. So over 1 million years, I can guarantee you that factory is not going to be still making laundry detergent. Nothing lasts that long. Nothing has that kind of stability. And yet that would be required in an origin of life kind of scenario, is to keep cranking out the same building blocks over time. So the take-home message here, and you're going to hear this take-home message a lot, and that is that concentrated building blocks, coming up with concentrated building blocks to form life, is a very unnatural kind of process. It just doesn't follow from physics and chemistry. So that is actually just the first step in the stairway. And now we'll go on to the second step, which is about chirality. And I assume most of the listeners have are familiar with chirality, basically, if you have a carbon atom and you attach it to 4 different bonds, 4 different things are bonded to it, then it'll have a chiral property. So it can either be left-handed or right-handed. And if you form molecules like this in some kind of lab, you're always going to get a 50-50 mixture of left hand and right hand, because these are equal energetics here. So you get a 50-50 mixture, and it's called a racemic mixture, and that is what natural processes do. So if natural processes are responsible for life, I would expect a 50-50 mixture in life, because that's natural. And in fact, the reality is dramatically different than that. So the amino acids that make up proteins, there's 20 amino acids. I think 19 of them have chirality, or 18. But each amino acid then has a chiral location. And almost without exception, in living things, those are left-handed, almost always left-handed. And in fact, if you get one amino acid with the wrong chirality, then the protein can lose its function. As said here, if only one amino acid is replaced with the wrong chirality, the protein will not fulfill its tasks, because you get destabilization induced by a distorted structure of the alpha helices and the beta sheets. And if you're not familiar with that, then maybe you don't know Jack Schitz, who's the author of this paper. Now we go to beyond amino acids, which you always hear about when people talk about chirality. Not many people talk about chirality of DNA. DNA, we're all familiar with the structure here. It's a beautiful structure, and we all kind of think, it must always have this kind of structure. It's quite complicated what's behind it, because you have a bunch of nucleotides all lined up perfectly. But each of the nucleotides, every single one of them, has 3 chiral locations. So you could have a left or right here, a left or right here, a left or right here. That gives you 8 possible chiralities in a nucleotide, in every one of them. And yet every one of them in life, including the 3.2 billion nucleotides in every cell in your body, has the exact same chirality, the exact same, not a mixture, but the same. And it gets worse when you go into RNA, because RNA has actually 4 chiral locations. And that means that there's 16 possible chiralities in a single RNA nucleotide. And yet every RNA molecule in life has the same chirality. And there's more. When you go to the phospholipids that make up every cell membrane, or big component of your cell membranes, they too have a chiral location. And in eukaryotes like us, or in bacteria, they are always left-handed. But rather unexplainably in archaea, the 3rd domain of life, they're always right-handed. And they're produced by entirely different kind of enzymes than we have in bacteria and eukaryotes. It's also interesting to know that when you die, that process of decay that we've gone through, you also have decay of this homochirality over time. You naturally move toward a racemic mixture as a living thing degrades. So you get back to 50-50. Why? Because 50-50 racemic mixture is natural. It's unnatural to have the homochirality, and our bodies are constantly fighting this battle and maintaining homochirality against all natural processes. So back to our conclusion here for this step is that homochirality is highly unnatural. It doesn't come from simple physics and chemistry. So that's the second step, and it's a requirement to get from chemistry to biology. We're now going to skip a step for the interest of time and go to one that's often ignored, is getting individual building blocks to link up in a perfect line. So back to DNA, you have this perfect line of nucleotides all linked up beautifully. And because DNA always looks like this, it kind of gives you the perception that that's just the way it has to be. But that's not true. Each nucleotide could take on different structure. In A here, I'm showing 2 nucleotides that are bonded together the way they are in DNA. So this is the right way to do it. But there are many ways to do it wrong. You could have this kind of bonding arrangement, which is wrong, and this, and none of these will lead to life. And so to think that you must have hundreds of thousands of nucleotides that are perfectly lined up with this chemical structure is very unnatural. And with each nucleotide you add to a growing chain, there's a chance you're going to get, oops, one wrong one jumps on the chain, therefore the chain is ruined and you're not going to get to life. So the probability just becomes astronomical of getting it right, astronomically impossible to get it right. So an analogy for this is to build a train. Let's say you want to make a train, and the train here represents either DNA or RNA or proteins. All these biopolymers of life are perfectly linked up like this, and extremely long, like, the simplest autonomously reproducing living organism has 580,000 nucleotides. So it's a perfect train with 580,000 boxcars lined up perfectly, where each boxcar is like a nucleotide, or in protein, it would be an amino acid for each boxcar. And the train tracks actually help you keep things in line. If you want to add a new car to the train tracks make it pretty easy to line up the cars and connect to each other. In life, that would be enzymes, big molecules, protein enzymes, which themselves are long trains of amino acids that are required to get this job done. And you also need energy, the energy to make the cars snap together, and that comes from ATP in life. So in life, this can be done, and it is done all the time with the help of enzymes and ATP to get the train all lined up. If you go to a prebiotic world, this would be the reality in the prebiotic world, is you have boxcars, the individual building blocks that you produce from like Miller Urey. But they're floating around. There's no train tracks. There's nothing to organize them. They're just loosely floating around. And you have to somehow connect these into a perfect line. And to make matters worse, there's some cabooses here, right? And when you hook up a caboose, that's the end of the growing train. There's no way to connect after the caboose, so it's the end. And you can also have a train that's growing in length, but the back of the train connects up to the front of the train and it makes a circle. Once that happens, you can't grow anymore. So that whole effort is wasted. And then to make it worse, as we've discussed already, that there's a whole lot of interfering junk molecules that are going to show up, like 36,000 junk molecules for every 80 that are useful molecules. So you're going to have a whole bunch of junk in here, and it's all floating around, and you're supposed to try to get 500,000 boxcars to connect and line up perfectly. You can just see how impossible that is. And this is a reality check on trying to make a biopolymer in a prebiotic world. Instead of hooking up to make a train, every time you're going to end up with a train wreck because you just can't get these things to line up perfectly. So the take-home message, once again, everybody's going to be familiar with this, is that long and perfectly arranged biopolymers are highly unnatural, very much unnatural, not something you're going to find outside of life. So we go on to the next step. And this is a step that we've addressed before on this channel, of the reproduction of biopolymers. So in order to have life, you have to have things that can reproduce themselves, right? And we know that replication of cells is an extremely complex process, and the origin of life people know that that's too complex to start there. So they talk about starting with self-replication of individual molecules like DNA or RNA. Particularly RNA comes to mind as the first self-replicating molecule. And that leads them to a concept called chemical evolution, which is where a molecule replicates itself. Occasionally there's some errors in making a slightly different copy of itself. And then the best replicators, those that can replicate the fastest, are going to win out, kind of like a natural selection process, an evolution process, and dominate and grow more complicated over time until eventually you have a living thing. That's the the sort of fairy tale that we're taught. Unfortunately, no one has ever seen a self-replicating molecule. You will see lots of claims made about this. Here's a book written by a chemistry professor in Israel, Adi Pros. And in his book, he says, molecular self-replication reaction is a reality, a reaction that actually does take place. And I find this extremely misleading. This could only be an accurate statement if you accept the most charitable and most generous definition of what self-replicating is. Just to make it clear, no one has ever seen a self-replicating molecule. A molecule that can replicate itself has never been found. 68 years since Miller-Urey, people have been trying, and nothing has been found. There is a lot of claims, like you saw with Adi Pros, but nothing actually that is truly self-replicating. Here's the dictionary definition of self-replicating: something that can reproduce itself by its own power or inherent nature, or it can reproduce itself autonomously. That to me is a common sense definition. Self-replicating, it can reproduce itself by its own power. But you'll see claims made like this. This is the first paper that came out about self-replicating nucleic acid, basically RNA, self-replicates. This is from almost 60 years ago. Saul Spiegelman, rather famous, came up with this. But when you actually read the paper, you find out that the RNA is not replicating itself. It is being replicated by an enzyme called Q-beta replicase, a very complicated enzyme that takes 1,200 amino acids. So think of a boxcar with 1,200, a train with 1,200 boxcars lined up perfectly with the exact sequence of a particular protein. It's actually 4 proteins combined together to make a Q-beta replicase, a very complicated enzyme that he had to add into the solution. And this is what's doing the replication. The RNA didn't replicate itself at all. And here's another claim, a more recent paper, where they call it a self-replicating RNA enzyme. And they say here that the RNA enzyme catalyzes the joining of oligonucleotide substrates to form additional copies of itself, undergoing self-replication with exponential growth. So this sounds like, they've got it. It's a done deal. We have a self-replicating molecule. But when you actually read the paper, you find out how they're stretching the definition of self-replicating to an extreme. What they actually did was they took an RNA molecule. This is like our train here, where we have boxcars. Each of these is a nucleotide lined up like a train, lined up like boxcars in a train. And it's a long train that goes all the way over here, and there's like 66 boxcars. Now, each one of these nucleotides we mentioned is homochiral, meaning they have the exact same chirality. Even though there's 16 possible chiralities for each of these letters, they're all perfectly homochiral, perfectly lined up to make a train. So this is a very unnatural molecule. It had to be produced from living organisms with some human engineering, some human intelligence in order to get this molecule in the first place. But this molecule, it's a long RNA, you call it a ribozyme because it's capable of performing a catalytic function. In the solution, they put a lot of this molecule in the solution, and they also put 2 other molecules in the solution. The other molecules happen to be 2 chunks of the same molecule. If you take the yellow molecule and you flip it vertically and then you flip it horizontally, you'll see that it looks like exactly what we have here. The blue plus green equals the yellow, just flipped upside down. And it's cool that the blue plus green has kind of the opposite nucleotides here, so that there's a pairing. These Watson-Crick base pairs form. And so naturally the yellow molecule attracts the green molecule here, and it attracts the blue molecule here and brings them into close proximity. And then they kind of cheated here by having a triphosphate, a nucleotide triphosphate here, which is a highly energized portion of the molecule that's really ready to undergo a reaction. When they come close together, a reaction happens and these two bond together, and then you make basically blue plus green equals yellow. You make another yellow molecule. So the yellow molecule has replicated itself, if you will, by producing one single bond in, two substrates. This is a very generous, charitable claim of what a self-replicating molecule can be. It's a lot like this. Let's say I take a car and I saw the car in half. It's a fully functional car, but I saw it in half, so now it doesn't work. But inside the car, I put some strong magnets and I put some mechanical clasping mechanisms in here. And then I take another car that's fully functional and I just push the back half of the car. I push the back half into the front half and you hear this clicking sound. It clicks together. And now you have a completely fully functional car from the two halves. And basically then if I were to jump up and down and call the newspapers and say, I have made the world's first self-replicating car, you think of what an absurd claim that would be, that we don't need car factories. We just let the cars reproduce themselves. It's an absurd claim, but that's exactly what they've claimed in that paper. So to summarize, there is no such thing as a self-replicating molecule. And self-replicating molecules would be highly unnatural. I mean, if self-replicating molecules like self-replicating RNA is actually the pathway to life, if that was the pathway to life via chemical evolution, self-replicating RNAs should be pervasive. They should be all over the place. Anyone who works with RNA should be like finding them all the time. Oh man, there's another self-replicating RNA. They're all over the place. That's the way it should be if RNA world and self-replicating RNA is actually the path to life. And that's because this process of chemical evolution would require millions and millions and billions of different self-replicating RNAs progressing forward, getting more and more complicated, leading to life through that process of chemical evolution. So self-replication should have or must have been prolific if that were in fact true. And yet no one's ever seen one. So what is the science telling us? It's pretty obvious. Now we're going to jump ahead a few steps for the interest of time. Now we're going to get to an interesting topic of repairing biopolymers. This one is hardly ever addressed. But when you have an RNA or a DNA or a protein, they fall apart pretty quickly. And the really strong example here is the COVID vaccine, right? Everybody's familiar with that right now. We're all excited about the COVID vaccine. That is an RNA molecule. And you've probably heard, how are they distributing the COVID vaccine, right? The Pfizer COVID vaccine has to be, shipped, maintained at much lower than freezing. It has to be at like minus 100 degrees. And why is that true? Because the molecule falls apart so quickly. Just spending 1 week at room temperature, it's useless. It'll fall apart. It has to be very carefully preserved because it's a weak molecule. So fortunately in life, we have repair mechanisms. Water, just water itself, UV radiation, oxidative damage, chemical attacks. We constantly have degradation of the RNA and the DNA in our bodies. DNA in every cell in your body is being broken apart thousands of times every day. Fortunately, we have a dozen or so mechanisms, basically protein machines, that go around and repair these problems. So one such repair mechanism, and this is like the simplest repair mechanism, is pervasive throughout all of life. It's called the base excision repair pathway, and it's shown here. And it takes one damaged base in DNA. This is like a straightened out DNA double helix. And you have one bad base, and you have an enzyme that extracts it, an enzyme that kind of opens up the DNA, an enzyme that prepares it, and then you have 2 enzymes to put back a proper base to repair it. So together you have 5 enzymes. Each of these is a complex protein on its own, and they together work together to get the job done. It's kind of an irreducibly complicated system to repair the DNA. And this is absolutely essential in order to have life, because the DNA falls apart so quickly. Now, the real challenge we come into here is that DNA repair is essential for life. That is a scientific fact. But the mechanisms in order to have DNA repair, which are these enzymes, where do the enzymes come from? Well, they are encoded in the DNA, okay? But you can't have the DNA unless you have the repair mechanism. But the repair mechanism is in the DNA. Does that make sense? How could this have started naturally? This is what's called Eigen's paradox. Anytime an information containing molecule like DNA gets to a certain length of, say, 100 nucleotides, it's going to fall apart unless you have a repair mechanism. But the repair mechanisms are going to take more than 100 nucleotides of DNA to be encoded in the DNA. So you can't get this to work. And this is one of a dozen different repair mechanisms in life. Now, because we have this sort of chicken and egg phenomenon here, a fancy term for that is causal circularity, right? You can't have A before you have B, but you can't have B before you have A. That's called causal circularity, and that is highly unnatural, right? Natural processes do not create things that have causal circularity. And this is just one example of that. We're going to see more. So that's the means for repairing biopolymers. Let me take a break at the moment to say that in this staircase, we have lots of required steps. I don't want to get into an argument over which step came before which step in the formation of life. I don't care what order these are in. The simple fact is that all of them are required to get from chemistry to biology. So some people might prefer a different order. That's fine. You can do it however you want. I just happen to do it this way for convenience. Next step is about the membranes. You know, every living thing, every cell has to be surrounded by a membrane to separate what's inside from what's outside, because the inside of the cell has to be a consistent environment called homeostasis. You know, no matter what happens to the environment outside, you can keep a nice, consistent environment inside, and that's required for life. So membranes in all of life are made up of phospholipids, which are kind of shown here. If you take phospholipids and you place them in water in just the right conditions, they will naturally form what's called a bilayer, like you see here. And this is, everybody knows this, I think. And this bilayer will curve up and form into a sphere. And so the origin of life people get really excited about this because they think here is one requirement, one step on the staircase that is easy. It's a slam dunk. And they get quite happy about that. But those people, and I see these claims all the time, people claiming that membranes are very easy to form and phospholipids are basically the same as cell membranes. They really don't know what they're talking about. They don't understand the complexity of any living thing's membrane, because any living thing has a membrane that's extraordinarily complex compared to this. Living things, all living things, even the simplest, require membranes that are active, meaning they're expending energy in order to move stuff across the membrane and doing it very selectively. So only things that they want to come in, come in. Only things you want to get out, get out. That's what membranes do in life. So one big example, an essential function of all membranes in life is to maintain an electrical voltage across the membrane. And this is done by a difference in the concentration of protons. You have more protons outside than you have inside. So that means the membrane has to be impermeable to protons, right? Protons can't get through the membrane on their own. They can't just diffuse across it. But that same membrane has to allow big stuff to get through, like building blocks for life, or nutrients, right? It has to come in, and we have to get the waste out, right? So the membrane needs to be really tight, but yet it can't be so tight that nothing gets in or out, or else it's just a tomb where you hold on to decaying, useless stuff. So how do you meet this conflicting set of requirements, a very tight membrane and a very open membrane? The only way to do that is with the use of proteins. So a true cell membrane is extremely complicated because it's filled with transmembrane proteins that have very specific roles of bringing stuff in that you need in, or kicking stuff out that you need out, responding to stimuli, keeping homeostasis within the cell. The simplest known organism that can reproduce itself autonomously is called Mycoplasma genitalium. It's a very simple bacteria. Even the simplest has 142 different kinds of proteins that it produces that have a job of operating within the cell membrane. So this little cartoon is still very simple compared to the reality. We should have 142 different kinds of proteins working here in the cell membrane, keeping things straight. Now, another interesting point is that no cell on its own is capable of producing its own membrane. I gotta say that again. No cell can produce its own membrane. Each cell inherits its membrane. And the cell is capable of augmenting it. It's capable of building more membrane onto its membrane. But no cell can start from scratch and produce a membrane. They don't even know how to do it, I would say. You inherit it. It comes from your parents. Now, if the cell produces a new protein that is supposed to go out into the membrane, how does a protein get inserted into this membrane? Because the membrane is rather tight, right? Well, it turns out you have to have a protein in the membrane in order to help sort of chaperone or escort a new protein to get across and be stuck within the membrane. So this is another kind of circular causality, that you have to have protein in the membrane in order to get a protein in the membrane. So to summarize this one, you've heard this line a few times, but here we go again, that active and selectively permeable membranes are highly unnatural. It's a very unnatural thing to find. Okay, just two more steps, because we're going to skip the top one. We need a means of harnessing energy. Energy, of course, is absolutely essential. And cells have to do it in a very particular way. Life, all of life requires, this is some thermodynamics here, all of life requires very high enthalpy in the molecules of life and very low entropy. So very organized molecules that are in a high energy state. This is highly unnatural. I'll say it again, it's highly unnatural to have this. Any chemical reaction that happens spontaneously would be the opposite of this. You don't have spontaneous chemical reactions that drive high enthalpy and low entropy at the same time. It just doesn't happen unless you have a way of harnessing energy through a thermodynamic engine. In other words, energy that's applied very specifically to do a certain task. Not energy like an explosion, which is very non-specific, right? Or just radiation or heat. You need something that applies energy very specifically. So in life, we have something, and this is across all of life, we have a process called chemiosmotic coupling. And what this does, basically, it takes your food and various sources of food for various kinds of life and converts it into a voltage across the membrane, right? You have more protons outside than you have inside. That's the voltage across the membrane. And that's a very complex process. We'll show a little more about that. And then that voltage is used to convert ADP, adenosine diphosphate, into adenosine triphosphate. And this is a lot like charging up batteries. So your body and every living thing is constantly converting ADP into ATP. And then the ATP, which is the charged up batteries, are used for all kinds of purposes to run molecular machines that do very specific applications of energy to keep these thermodynamics going. To give you a little feel for the first part of it, this is how do you generate the potential across the membrane. There is the respiratory complexes. This is complex number 1, and it's made up of 16 different proteins, unimaginable complexity here. And it lives in the membrane, as you can see here. And this thing actually strips electrons from the food, whatever food you've got, strips those electrons. You have an electron transport chain, a very delicate process of handling hot potatoes, the electrons coming through here. And as the electrons are stripped off, you have protons being pumped across the membrane. So you have fewer protons inside the cell and many protons outside. This is constantly pumping protons out against the diffusion gradient here. And this is one of 3 respiratory complexes that work together to get this job done. And together they require 25 different proteins of great complexity just for the process of pumping protons across the membrane. And that's incredibly complicated, and it's in all forms of life in one form or another. So pumping protons across the membrane is awesome, but it doesn't do anything unless you can make use of this voltage gradient. And that would be what ATP synthase does. It harnesses the energy of the proton gradient. So we have few protons inside, many protons outside. This is an electrical voltage which drives this electric motor. ATP synthase is made up of 20 different proteins, and it's a rotating shaft driven by this voltage. It allows the protons to get into the cell only if they drive the motor around. And by driving the motor around, we convert ADP into ATP. We're charging up those batteries. It can spin at 7,800 RPMs. So if you combine the complexity of this with those respiratory complexes, we're looking at 45 different proteins that are required to generate ATP through that process of chemiosmotic coupling. I hope I'm not losing people with all this complexity here, but just to appreciate that this is in every living organism, and just generating the potential across the membrane does nothing for you unless you have this machine to make use of it. It's kind of an irreducibly complex system. You got to have all of it together because just having a few pieces will get you nowhere. And that's how you take energy and apply it very specifically to build up the cell and to keep life going and to maintain life. Even those DNA repair mechanisms that I talked about, they need this ATP energy. Everything needs energy. Okay, so we have one more step I'm going to go over, which is the interdependency of DNA, RNA, and proteins. So the 3 main biomolecules of life are hugely interdependent. You can't have one without the other. So that creates a big chicken and egg problem of which one came first. There are so many of these examples, chicken and egg examples in life, which the fancy name is causal circularity. But we already talked about 2, that the DNA repair mechanisms can't exist without the assistance of DNA repair mechanisms. And we talked about how proteins getting into a membrane, proteins in a membrane are required in order to get new proteins into a membrane. I mean, there's many more examples of this, there's hundreds of them, but let's just do a few. In order to replicate DNA, you've got to replicate DNA in order to replicate a cell, right? But in order to replicate DNA, you need a minimum of 25 very specific proteins that come together to make 14 enzymes. And that's the minimal set required in order to replicate DNA. And it's believed that those proteins evolved. Well, how do you evolve if you can't replicate, right? Evolution requires replication, but you can't have replication until you have these 25 proteins. So they can't evolve because you can't replicate. That's a chicken and egg problem. It's essential if you're going to make a protein that you already have proteins. And that's because ribosomes are the things that manufacture proteins. And a lot of what constitutes a ribosome is protein, but the ribosomes make the proteins. So how do you get the first ribosome? That's a chicken and egg problem. If you're going to make a protein, you have to transcribe DNA into RNA. And what does that is a protein, which is called RNA polymerase, right? But RNA polymerase, which is a protein, had to be formed by transcription. But the transcription is done by RNA polymerase. So how do you get this started? There's another chicken and egg. Ribosomes also have an essential component of RNA. RNA is an essential component of ribosomes, but the RNA comes from a protein. The protein comes from the ribosome, but the ribosome has to have RNA. So once again, chicken and egg problem. And there's so many of them. So back to what I said before, causal circularity is highly unnatural, and it's prolific within living things. So because of these things all being interdependent, the stairway to life is actually kind of the wrong metaphor. It's really more of a cliff that you would have to have all of the things all at once in order to get it done. Not just steps, but you got to have all of it at once like a cliff. Now, I wanted to address one other point here, and that is very often you hear the term protocell. People use this term to try to fill in the gap, because getting from chemistry to biology, that big staircase, the big gap there, everybody knows that's a problem. And so they start to use the term proto to sort of fill in the gap. You could have proto-RNA or proto-DNA or proto-proteins or proto-membranes. And often you hear the term protocell, which is supposed to be a really simple, basic cell that got things started. And this term really kind of gets my blood pressure up because it makes no sense. The smallest, simplest living organism today, we already talked about, Mycoplasma genitalium, that can reproduce itself autonomously. It has 516 genes and almost 580,000 base pairs of DNA, okay? This is the simplest organism around today. And people imagine that there are simpler organisms in the past, protocells, which is what got us to where we are today. So these are imagined to bridge the gaps. And this doesn't make any sense because, well, let me start off by saying people are trying to simplify life beyond Mycoplasma genitalium. A lot of people are trying to knock out genes to see what is the simplest life that can reproduce itself. And the best effort to that end, I think, was Craig Venter. He recently reported, the simplest possible cell with 473 genes and 513,000 base pairs of DNA. And what he found is that as you start to knock out genes, you may be able to still have a living thing, but it becomes very dependent upon a coddled environment. You need an environment that hands everything to you on a silver platter. It's kind of like this, can a human being live without legs? Sure, you can live without legs, but you kind of need help. You need an environment that's going to make life a little easier. Can you live without arms? Can you live without eyeballs? Yeah, you can, but you need more and more help, right? And that's what's going on as people try to knock out genes. And the whole process to me doesn't make sense. And that's because life on its own is constantly working to try to simplify itself. Okay. You may know Richard Lenski did this experiment with E. coli, with 70,000 generations of E. coli in his laboratory. And he found that after only 2,000 generations, the E. coli in all 12 flasks completely ejected the operon to metabolize ribose. Now, let me explain that. So ribose is a sugar. Ribose can be used by E. coli as a source of food. But in the environment that Lenski gave these E. coli, there was no ribose. They had plenty of glucose to live on. Or at least that was the main source of food. So the bacteria, by random mutations, dropped an entire chunk of DNA that was there to metabolize ribose, and they could survive better. In fact, they could reproduce about 3% faster because they got rid of the baggage of this unnecessary DNA. and they did better. So they're always trying to simplify themselves. They're trying to reduce their genome to be the best surviving they can. This is a natural process. So having humans come in and try to chop out pieces of DNA isn't going to be productive because it's already happening. So unless you provide a very coddled environment, we're not going to get any simpler than what we already have. And a very coddled environment is not consistent with prebiotic situation, right? You can't have everything provided for you in a prebiotic situation. So I'd like to say very strongly that protocells are imaginary. They're an imaginary entity that are used to fill in the gaps. It's kind of a materialism of the gaps approach, and they make no sense and they don't exist. Okay, so back to the full stairway. We're stepping back to the big picture now. And we know that gravity, the natural process of physics and chemistry, drag us down the stairway as we die, as we decay. And yet some people believe that the opposite is true, that you can ascend the stairway. To me, those people believing that you can ascend this stairway are well depicted by this cartoon. It says here, for years, he had spent every free moment working on his perpetual motion machine, and so far he was up to 18 seconds. People that are trying to convince us that life began through natural processes are essentially working on a perpetual motion machine. And they can make a little progress. Maybe they can get it up to 20 seconds. And they can pound their fist and get a lot of media attention that they're making progress. But in the end, it's a waste of effort. We know enough about science. We know enough thermodynamics that we know perpetual motion machines are not natural. It cannot be obtained by a natural process, so don't waste your time trying to build a perpetual motion machine. And the same is true about a natural origin of life. We know enough, as I've shown you in the staircase and all the different examples. We know what natural processes can do. We know thermodynamics and chemistry and physics, and we know that you can't have a natural origin to life. And yet, there will always be people who believe. And so you have all these people that we talked about at the beginning and our high school biology textbooks that still believe. So I'm going to diverge for the last 2 minutes here, just talk about some philosophy of why these people believe this. And I think this analogy helps. Let's imagine that you have a dead body appears. Somebody finds a dead body. Nobody knows where the dead body, how they died. Nobody saw it, no witnesses. So, of course, the coroner comes in. The coroner is trying to figure out why the person died. And at the highest level, the coroner is trying to answer a question. What is the cause of death? And trying to answer that as two parts. Either it was a natural cause of death, like cancer, heart attack, stroke, or it's an unnatural cause of death, like a murder or a suicide. So to answer that simple question, which one of these is it? They're going to look at the evidence. And for the evidence, in this particular made-up case, there's a single wound above the left ear of the deceased. And when they open up the skull, they find extensive disruption of the brain tissue. And they recover a single foreign object. And it was composed of lead and copper. And it looks like this. And of course, any coroner would know that this used to look like that. That's how it got started. Now, this evidence seems pretty clearly to point in the direction of an unnatural death, right? I hope everyone will agree with me. Unfortunately, this coroner has a very strongly held belief. This coroner believes that under no circumstances ever has anyone died from an unnatural death. It's just not possible. That's what they believe. And so they see the evidence here, and they're trying to get it to point to a natural death because that's what they believe has to be true. So they dig a little deeper. They're trying to explain this. And they find out actually that the body was found in the vicinity of a volcano, an active volcano. And they think, aha, now I can explain it. The volcano spit out this foreign object, which unfortunately ended up in the head of the deceased. And so the cause of death was a volcanic eruption, a natural cause of death. Now, if you think that this is a bad coroner, if you think this is bad science, I agree with you completely. But I ask you then to ponder this question of how did life first begin? Was it, and there's a simple two-part answer here. It's either a natural source, simple physics and chemistry doing what they do, or did life begin from a supernatural cause? So let's look at the evidence, right? The evidence is everything I've been presenting for over the last hour. You have homochirality, which is very unnatural. You've got cell membranes of extreme complexity that are active and selectively permeable, very unnatural. You have the energy harnessing process. You have protein repair. You have, DNA repair processes. All of these chicken and egg circular causalities going along. And in fact, the entire stairway to life, all of these steps. Each of these pieces of evidence strongly points away from natural and strongly, therefore, points in this direction. And that's what the science says if you're free to follow the evidence where it leads. Unfortunately, we are in a society where science is defined as only being able to address natural causes. Science is defined in a way where it can only speak to natural causes. In other words, this cannot be considered in a part of a scientific investigation. So, rather than thinking what is actually the answer to this question, the scientific method as we've defined it is seeking the best natural explanation, and that's all that's allowed. In other words, we've constrained science so that it's not actually capable of getting to the truth. It's blocking out one of the possible explanations, and it's only possible to obtain a natural cause because that's the way it's been defined. And so the conclusion that you hear in newspapers and media, in many scientific manuscripts, is that life started naturally and the case is closed. And that's because we've started off by blocking out the other possibility. And what I think is even worse here is that in our schools, the blocking of this possibility is actually concealed. It's not like they explain to the students, hey, it is possible that there was a supernatural cause to the start of life. But for now, we're just going to think about natural causes and we're going to do science focusing on natural causes. They don't explain it that way. All they say in the schools is that science encourages a belief. They're going to say science points to a natural explanation for life. That's what they're teaching our students. And I think hiding this is actually making things much worse. So, wrapping up here, why do people believe in abiogenesis? Well, science has been defined in a way where it can only point to natural causes. Science is not really concerned with finding the truth. It's concerned with finding the best natural explanation. And I think another reason why people believe in abiogenesis is that atheism has no rational basis unless you have a natural cause for the beginning of life. You've got to have abiogenesis in order to be a rational atheist. You can certainly be an atheist just because you feel like it, but you probably won't have that comfort of knowing that there's a rational basis. So I end with this quote from Harold Urey of the famous Miller-Urey experiment back in 1952. He said, "All of us who study the origin of life find that the more we look into it, the more we feel it is too complex to have evolved anywhere. We all believe as an article of faith that life evolved from dead matter on this planet." So he said it's an article of faith, and I completely agree with that. It's an article of faith. And thank you all for listening for a long presentation. And that's all I have to share, and I'm happy to discuss and answer any questions.
Wow, I have to say, Rob, that was an absolutely amazing presentation. We've got a lot of kind words in the chat. We've had over 30 people the entire time. So you've kept everybody's attention. Looks like we got 35 people right now. Lena says, legendary presentation. Sal says, bravo, Rob Stadler. This is the best ID-friendly presentation for biology ever. So, and I want to point out those slides, Rob, I loved them. Great visuals, kept my attention the entire time. Very impressive. So great work. And I love, I'll say, that just officially put an end to abiogenesis and naturalism. You know,
I wish it were so.
And I love your materialism of the gaps point. You know, that is a lot of what I see personally in terms of their rescue devices, especially with this imaginary protocell appeal that you covered in your presentation for a bit there. I do have a ton of questions, actually. We've had a very lively chat, but I want to hand it over to George. George, if you had any comments, brother, go ahead.
Yeah, well, Rob actually had me in stitches with not knowing jack shit. But I have to say, Rob, I don't know jack shit, but I know his brother, chicken shit. No. For those that are listening, look, I ordered the book some time ago, but they told me that it would take a few months to get to me. So I ordered the Kindle version. And for those that haven't read it, please do so. There are so many other things that Rob hasn't mentioned that are in the book that will really open up your eyes. I was surprised, actually. We all know about the DNA repair system, but reading the book, I was surprised that there's actually so many specific or a variety of specialized DNA repair mechanisms to counter so many of the daily attacks on our DNA. And I'll just mention some of those. The base in excision repair system, the nucleotide excision repair, the homologous recombination repair, the mismatch repair, the photo reactivation, the non-homologous end joining, the translation synthesis and processing by the mRN complex. I never knew about any of these. So I urge people listening, get the book, read it, and you'll be amazed at some of the information that's in there.
Amen. Amen. I couldn't recommend it more. It's a phenomenal book, Rob.
Thank you. And you know, it's what's astounding to me is you see this this conflict between belief in RNA world hypothesis, which is that over millions of years, RNA molecules became more complex through self-replication and led to life. And yet here we are with the COVID vaccine, and the COVID vaccine being an RNA molecule that has to be kept at minus 100 degrees or else it falls apart. You know, it can't survive at room temperature for like a week. And yet we're supposed to believe that RNA, you know, bumbled around on the earth for 100 million years, becoming more and more complex. So we have life. Come on, people, wake up.
Yeah,
well,
Rob, I've heard numerous biologists tell me that I won't name them, but they say that biology is equal to chemistry plus energy. But what they're forgetting is the many layers of information that are embedded in there. So they have to explain the origin of that information. And if I can quote Werner Gitt, most people probably know Werner Gitt. He says the grand theory of evolution would gain some empirical support if it could be demonstrated in a real experiment that information could arise from matter left to itself without the addition of intelligence. Despite the most intensive worldwide efforts, this has never been observed. So they have to figure out where that information came from. And we know that information can only come from a sender, an intelligence.
And I think you know that Perry Marshall has this $10 million prize for someone who can demonstrate what you just suggested as a challenge. Yes.
Standing, I don't know if you caught most of the questions, but I caught some of them. Do you want me to ask one question that logical plausible, probable, our dear friend John Maddox asked.
Sure. Yeah. Go ahead.
Yeah, John asks, you mentioned almost all amino acids are homochiral. Are there exceptions in DNA/RNA, or is it exclusive?
So DNA and RNA made up of nucleotides, those are all chiral molecules, right? They all, like DNA always has 3 chiral locations, RNA always has 4 in every nucleotide, in every living thing all the time. There's no exceptions for that. But in amino acids to make proteins, I believe alanine is the most simple amino acid, and that is not a chiral molecule because it's very simple. It doesn't have 4 different attachments to the central carbon.
Yeah, he follows it up by saying, do cells separate chirality or create it?
I think both is the answer.
Okay.
There are special enzymes called epimerases or racemases that are enzymes that living things produce that specifically change the chirality and enforce homochirality. And of course, interesting to say, those enzymes themselves are homochiral, right?
Yeah. Over to you, Standing.
Yes, I've got a question here. Godzilla Freak question. How would you respond to those who say that future research will find how abiogenesis can happen, which I've personally heard over and over and over again by the critics?
Yeah, you clearly see that. That claim is made constantly. I think I have a simple response to that, and that is that, and I think you saw in the beginning of my presentation, back to the Miller-Urey experiment, that's 68 years ago. That experiment really kind of kicked off a lot of research, a lot of interest in origin of life. And in those 68 years, the goalpost, right, which is producing a living self-replicating cell, has pulled away exponentially because we've learned so much about the complexity of life in 68 years. If you look at the staircase with those 12 steps that I talked about, 68 years ago, I don't think we didn't know 11 of the 12 steps existed, right? We hardly knew what DNA was back then. And so back then there was only one step, and they thought Miller-Urey pretty much took care of that step, so that George Gaylord Simpson was pretty convinced that it was a done deal. In 68 years, we've added 11 steps, and I'm sure there's actually more than that. And the progress in laboratory efforts to make life has been extremely minimal compared to this exponential growth in the goalposts, right? And I can predict very strongly that the next 100 years is going to do exactly that again and again and again. Even in the simplest form of life, and this is from Craig Venter, in that, he tried to simplify life. He found that 20% of the genes in the simplest form of life he came up with, 20% of the genes, we still have no idea what they do. We don't know what they're doing. And I can guarantee you that when we figure out what they do, it's going to add steps onto the staircase. And that is also just looking at the DNA information. And we know that life has a lot of epigenetics, a lot of information above and beyond DNA that we still haven't explored, like the sugar code and histones and all. There's so many codes in there that we haven't explored yet. So we're going to be adding a lot of stairs onto the staircase. Meanwhile, the origin of life community will still be back at step number 1, trying to find a way to make the building blocks of life and concentrate them.
Well, that's a great response. That's a really good response. Yes, the more we discover about the complexity of the genome and the DNA molecule is only going to add steps while they're still stuck on step 1. And I really enjoyed your entire presentation, but I like how you pointed out the chicken and egg problems that abound, right? Chicken and egg problem after chicken and egg problem. You touched on the DNA repair enzyme and how it's essential for life, but the mechanism for DNA repair are encoded in the DNA, right?
Yeah.
So how did this have started naturally? And one analogy that I liked earlier on in your presentation, Rob, had to do with the Thanksgiving dinner. You've got your mashed potatoes, your stuffing, your gravy, your apple pie, and you got to make everything in isolation, for example. But with the origin of life and all these experiments, and a real origin of life situation, if it happened, you have to make your entire Thanksgiving dinner in one single pot. So isolation isn't going to work, as you pointed out, which these abiogenesis experiments tend to actually do.
Yeah, they're all in isolation like that, and they claim victory for some little progress made in isolation. And they expect people to kind of glue together those little success stories into believing that the whole thing is a possibility.
Right. Well said. Well said. We've got our brother Sal here, Evidence and Reasons. Anybody who has not yet subscribed to Salvador's channel, please do. He's putting out some great content. His question for you, Rob, he's curious, any more books or videos in the works?
I hope so. I'm hoping with the group to produce a series of animated short videos that explain this kind of in lay terms. It's hard to do a presentation like this because you don't know the level of the audience that you're trying to reach. So you got to try to reach people in all different levels, but we want to make kind of animated short videos that explain it to the lay audience. And that's been a big project for now.
Rob, one of the things I find amazing with biology books, when they quote the Uromilli experiment, they always tend to say they were successful in producing 2% of that mixture as amino acids, but they never tell you that it's racemic, that it's a 50-50 mix, and that simulates death. It doesn't simulate life.
Right. Yeah, it's amazing. If you're out for a walk in the woods and you come upon on a rock somewhere, you come upon some amino acids, you could actually take them to the lab, and if the amino acids are homochiral, then they came from a living thing. And if they're racemic mixture, then they don't have anything to do with life. So that's how definitive homochiral is in separating living from non-living things. And in the last 20 years or so, there have been some chemical processes that can move racemic mixtures toward homochirality. I don't think they can even get there, but they can get to maybe 90% of there. The most famous, I think, is the Soei reaction from Japan. But when you actually look at the chemistry behind that, it's extremely not prebiotically plausible, right? The kind of chemistry that goes into that is some nasty, very unnatural chemistry that can, it can move you toward homochirality, but can't get there. But somehow that kind of recent science is enough to convince some people that homochirality is not a problem anymore. But again, they're just so willing to accept what they find. Like, no one, I think, has talked about the chirality problem with DNA and RNA, which, as I explained, is much more difficult than with amino acids.
I've got a question here, Rob. I can't put it on the screen, unfortunately. This was from earlier on in the in the lecture. It's from Brandon, and he asks, for the skeptics that haven't read his book or page 214, can Dr. Stadler refute the silly God of the gaps argument? Oh, it's so hard to do, therefore God must have done it.
Yeah, good question. Thanks. I get that a lot. People say, "Oh, you're just doing God of the gaps argument all the time," and so God of the gaps is basically saying, I have no idea what's going on here, and therefore God did it. So you're filling in the gap of lack of understanding with God, and so people often claim that that's my approach here. We don't know what's going on, so it must have been God. And I'd like to say, really, it's the exact opposite of that. That we know enough. I've been trying to say this in the talk, is we know enough about biology. We know enough about the minimum requirements for life and what living things have in their complexity. We know enough thermodynamics. We know enough chemistry. Bringing that all together, we know enough to know that natural processes cannot have accomplished this. So there's no gaps. There's no hand waving here. It's that we now know enough to say that natural processes can't do this. And then I think the opposite for those who believe that life started naturally. I think that they're using a materialism of the gaps, basically saying, we know that natural processes tend to degrade life over time to simple molecules, but I believe that simple molecules through natural processes complexified to become life. I have no idea how that could happen, but I know it was a natural process.
Right.
That's naturalism of the gaps.
Right. Naturalism and materialism of the gaps. Great response. I apologize, George, if you had a question, maybe right after this one. This one just came in and I can at least pop it up on the screen. Question from Praise I Am That I Am. What abiogenesis hypothesis, in your opinion, Rob, is more absurd? The primordial ooze or ocean vent hypothesis?
Well, this question takes me on a little tangent, and I think you had John Sephardi on your show at some point, and I remember he mentioned that one of my favorite papers on this too. There's a paper on abiogenesis, and I think it's titled like the RNA world hypothesis, the worst hypothesis for abiogenesis, except for all of the others.
Yeah. Oh yeah, I remember.
That was actually the title of a scientific paper. So to try to answer that question, I think they're all the worst. I can't. I mean, I don't have a favorite. When you talk to people who support abiogenesis, they all have their favorite hypothesis, and they'll rip on everybody else's hypothesis. But
right. Yeah, they're all debunking each other because they're all the worst, essentially.
Yeah, right.
One's just worse than the other. George.
Yeah. I've got a question here from Hate Love Nothing. His question is: are there any current technologies that are capable of altering the way in which the information is handled? Would this change anything, or is the information not tangible?
Are there technologies that change the way information is handled in living things?
I'm assuming so, yeah.
Yeah, I just read yesterday about how one of the COVID vaccines works, and I found it quite fascinating, really. But so it's an RNA molecule, but somehow if you inject RNA into the body, and it's the normal kind of RNA that has uracil as one of the nucleotides, it's much more likely to be degraded, that the body will attack that and break it apart, and your vaccine will come to nothing. But the intelligent engineers that made this vaccine, they actually replaced all of the uracil in the RNA with something that's very close to uracil, but it's kind of like a masquerade, a slight change to it. And somehow the body doesn't recognize that, and then it allows it to pass through and get to the ribosomes and actually produce the protein, which is the intent of the vaccine. So there, maybe that's kind of answering the question, that they're kind of tweaking the way the body is dealing with the information and fooling it in some ways in order to have the vaccine work. It's kind of fascinating.
That's a great response, Rob. I've got a question here from this is Tony. He's helped us out with some of these interviews as well in the past. SFT, can you ask Rob about the highly manipulated purification processes that chemists employ to isolate their amino acids? I know this involves pH manipulation, etc.
Yeah, if you want to get into details, I'm not the right person to get into, how you do separation and purification, but we could phone a friend maybe. But I know that it's essential for, it's essential to purify and to filter out all the junk that I mentioned and get to the one thing you have. And that's done in these manuscripts. And they don't explain how that could have happened in a prebiotic situation, but they do it in the laboratory, and that's a kind of cheating,
Right.
That takes you away from reality. I've never seen anybody try to explain that in a prebiotically plausible situation.
Right. Right. I completely agree. One thing I like about your book too, I mean, so many the things I like about it, but I like the ending where you address some of these anticipated objections, right? Because a lot of times it's the same talking points over and over again that are being employed. And you mentioned manuscripts there. And my question has to do with a question that you did answer in this section, but it's because I constantly hear it all the time in discussions, debates, or responses to interviews and lectures where the critic will point to, as a form of an argument, they'll point to the 100,000 peer-reviewed scientific manuscripts that have been published on abiogenesis as some kind of counter rebuttal, maybe insinuating that we are ignoring some of these papers that apparently have given evidence or credence to abiogenesis. What's the best way to respond to that, I guess, is my question, Rob.
So, good question. Unfortunately, we're in a situation where the literature is extremely biased. It's biased because only one answer is publishable, right? This gets back to when I put the no smoking sign on anything supernatural, right? So science has defined that the science can only address natural causes, right? And so only natural explanations are considered science. Only natural explanations can be published. And frankly, we all know the bias of scientific manuscripts, that positive results are going to get published and negative results don't tend to be published. They tend to be thrown away. So yes, you have thousands of manuscripts in the scientific literature, peer-reviewed literature about abiogenesis that are encouraging. They support natural causes because they have to, and they're positive generally because that's what gets published. So there's an incredible bias to that, and there's no balancing out there. So in writing this book, I'm attempting to provide just a little bit of balance to this, because otherwise you get a one-sided story. It's almost a propaganda, and that's all that can be published. And you also have the problem of the media. Then the media comes in and they hype it up, right? They're always kind of hyperbole applied to what they see. And they make it sound like it's a done deal. Everything's been explained. We have self-replicating molecules, on and on and on. And the media needs to take some responsibility, I guess, for falling for the clickbait kind of hyperbole that they generate a lot.
Right. A lot of it is clickbait. You're absolutely right. And, I can't thank you enough for all the amazing work you've done in not only this book, but your previous book on evolution. And a lot of the people in the audience today, they look like they have read your book. And they own your book. So, but I want to point out to anybody in the audience who may not have read it yet, I've got both of your books, Rob, in the description box of this video, as well as our other discussions on these topics. So please look into it. It's a must read. It's a must have. Personally, my last question, and then I'll hand it to George, and we'll start winding it down, Rob, because you've given us almost 2 hours of your time, and I can't thank you enough. I hear this all the time as kind of a counter rebuttal or lack of, in that, we are hindering further research and possible advancement on origin of life, on abiogenesis. They're saying, okay, maybe we don't have an answer now, but what do you want us to do? To just stop? And I believe you actually do answer this in your book as well. I'm curious as to your thoughts, because I personally have heard that a lot, that we're hindering scientific advancement.
Mm-hmm. Well, two things. First is I can think of thousands of things that science could be applied to that are more beneficial for society, right? I mean, if you have $1 million to spend on science, you got cancer, you got COVID, you got, all kinds of problems that society faces that science can improve our lives and make things better. So I would hope that the vast majority of the money goes to those kind of useful things and almost nothing goes to abiogenesis, right? Because it's a waste of money. But the second thought that comes to mind goes back to what I mentioned in the talk, is that we don't, as a society, we don't invest any grant money that I'm aware of into finding a perpetual motion machine, right? Because we're smart enough now to know, we know enough science to know that that is a waste of money. Maybe there's some crazy rich person who's investing in it with his own personal funds. I don't know. But in general, hopefully our government and big agencies are smart enough to know that that is not a good use of research money. And in my strong opinion, any money dedicated toward abiogenesis research is very similar to that. It's not productive for society, and it's basically throwing your money down the hole.
Amen. Amen. Well said. I really wanted the audience to hear those words. That was a great answer. It's a waste of money, and what you said really stands out to me in the fact that the money is far better put into research programs on cancer, fighting disease, the COVID, everything to do with COVID that we're experiencing right now. There's far more important things for us to study and research than abiogenesis, which, as we've seen today, it has been dismantled, has no evidence for. So great response. George, I'll hand it over to you if you have some final words or a couple final questions.
Yeah, I've got a proposition regarding consensus science. It's a 4-step process. Step 1 says every scientist accepts evolution is a fact. Step 2, one scientist comes out and says, I don't believe evolution is a fact. Step 3, the guy is censored, thrown under the bus, loses his funding, he's sacked. And step 4 now says, now every scientist accepts evolution as a fact. That's consensus science for you.
Right, that's brilliant.
But I've got one question, Rob. Before we finish off, I read somewhere about a 4-stranded knot of genetic code. Apparently, it's been controversial among scientists for years. There was a journal, Nature Chemistry, provides the first direct evidence that it does exist and that it may play an important role in regulating our genes. Now, I'm always careful to accept something when it says may. Can you expand on this if you're aware of this paper?
I'm aware of it in that I've just heard about it. Maybe I read the abstract, but I can't speak to the details of it. But we certainly are finding over time, we're learning more and more about layers and layers of information in DNA that's beyond just the simple one-dimensional code that's contained in there. That the way the 3-dimensional structure of it and the way some parts of DNA come in proximity to other parts. And as you said, at some points you can have actually instead of a double helix, you can actually have a quad. That there's so much we need to learn, and this is a good use of money, right? This kind of research to understand these things. I was just reading today about how we in prokaryotes, they don't have organelles in the single cell bacteria and so forth. There's not organelles. But now they're thinking that you have what's called condensates, which is pockets where a bunch of proteins come together, and it's kind of like a drop of oil in water kind of thing, where you have a condensate that is very important for the function of that cell. And we're just beginning to learn about that you have areas that are kind of membraneless organelles. Anyway, I'm getting off on a sidetrack, but there's so many things we still need to learn, and that's a very good investment of money because understanding this stuff will lead to disease cure and so forth. That's very important for society. So let's put our money into that.
Yeah, I was at the grocery store the other day, Rob, and I asked the store worker there. I said, "Are these genetically modified eggplants?" And the store worker says, "Why do you ask?" And immediately after that, the eggplant says, "Yeah, why do you ask?"
I don't know where
sorry,
but keep it up, brother. Keep it up.
It's good to have some levity in an otherwise heavy topic. So thank you, George.
Yes.
Yeah, a bit of relief. Yeah,
Lightens it
Good
up.
on you, Rob. We
Oh,
all love you, Rob.
Thank you.
Yeah, we love you. And this was the type of presentation where I'm probably going to make a coffee after this and re-listen to it and take some more notes, because wow, it was so informative, and the visuals, the slides, everything about it was awesome. I mean, we're going on 2 hours. We still have almost 40 people in the chat, so we could probably ask you questions for 10 hours, but we're not going to do that to you, Rob. You've done an awesome job today, and we appreciate your time, and this was awesome. I want people to share it around, please. We want to get this information out to as many people as possible. And once again, thank you, Rob. I want to give you the floor for some final words, some final thoughts before we close it down.
Well, thank you. Thanks for your time, everybody. I think you saw in the last slide I presented, there's an email address for me. You can send questions that way or contact me. So I appreciate that. And thanks everybody. Remember that life is just chock full of very unnatural things that clearly separate, and this is very different than, years ago, there was what was called vitalism, which was a belief that, life fundamentally was constructed of different molecules and different processes than non-life. And that, of course, went away as we found, through reductionism, we found that the molecules of life, the atoms and so forth, are really no different than in non-life. But I think we overdid it. I think the pendulum swung too far, in that, as you saw in my presentation, there really are some very special things about life that we need to appreciate, that separate it from non-life, such as the homochirality.
And Rob, I'd like to finish off by wishing you a happy new year. Hopefully 2021 will be a better year, and God bless you.
Thank you. God bless you guys. Have a good evening.
Well said. God bless. Have a good evening. And everybody in the chat, God bless you. And we'll see you tonight. Okay. SFT is out.